In this video, we'll talk about the structure and function of enzymes. We'll also talk about enzyme inhibition. So enzymes are biological catalysts. They're also functional proteins that speed up reactions without actually getting used up themselves. They provide an alternate pathway with a lower activation energy than the uncatalyzed version of the reaction. They're extremely specific for their substrates, and a few fun facts about enzymes: they usually end in A's, and the root word is usually related to the substrate. So for instance, lactase would digest lactose. Carbohydrases would digest carbohydrates. So activation energy is the energy that's required to start the reaction, to allow it to happen. So if you look here on this graph, your enzyme and your substrate causes a much lower activation energy. So it provides an alternate pathway than if there was no enzyme at all. So if you recall, metabolism is the sum of all the anabolic and catabolic reactions within the cell or the body. And remember, anabolism builds up macromolecules from their monomers through condensation reactions, and that consumes energy. And the hydrolysis breaks macromolecules into their component parts through hydrolysis, and that releases energy. And you can see the two reactions taking place here. So hydrolysis puts water in to split them up, and condensation reactions takes water out to build them together. So we're asked to look at one beneficial enzyme, and that's PETase. It's a naturally occurring enzyme that actually has the ability to break down polyethylene terephthalate, which is one of the most common plastics, the ones that you would find in like plastic water bottles. And it can do that within 24 hours. And PET plastic makes up 12% of the plastic worldwide. So this is a really beneficial discovery, because without it, it could take up to 500 years for that plastic to degrade. So what's a substrate? That's the substance that the enzyme itself acts on to speed up the reaction. And it will bind to the enzyme at the active site. So this is our enzyme. This one's called hexokinase. And the substrate is glucose. So it inserts itself here at the active site. And then it forms something called the enzyme substrate complex. And then the reaction takes place. So the enzyme converts the substrate into products, and then it'll release the product away, and then the enzyme can then work on another one. And we'll talk about the fit, how they fit together in our next slide. So from chemistry, you'll remember the collision theory. Molecules are constantly in motion, moving throughout the fluid, and they rely on the collision model to have an interaction with the substrate, the enzymes and the substrates. Because the molecules are all in motion, they'll have to have an effective collision to actually work. So sometimes the substrate will be immobilized and then the enzyme is moving around, and sometimes the enzyme will be immobilized and the substrate will have to find it. So that's one limitation and one function of enzymes as well. So the old model for enzyme function used to be called the lock and key model. And basically, in this theory, the enzyme's active site was the lock and the substrate was the key. And it stated that the active site was the exact shape of the substrate, and then only the correct key would fit in the lock. And that theory was good in that it helped explain the enzyme specificity for its substrate, but the way enzymes work is a little bit more complex than that. So the model that we hold to be true now is the induced fit model, and it's kind of like putting on a glove. The glove isn't fully the shape of your hand until you put your hand into the glove. It takes the shape of the hand. So an enzyme's active site is the same way. It will mold to take the shape of the substrate once the substrate starts to bind. So you can see here that this isn't the perfect fit, but as soon as it starts to bind, it does mold and fill in all the spaces. The substrate will get converted to products and then will be released from the enzyme. And the enzyme's active site will go back to the original shape after the product leaves. So because enzymes are proteins, they can be denatured. And that will affect both the tertiary and quaternary structure of the protein. So the enzyme will unfold or unravel, and it won't be able to do its job. So there's a lot of things that can affect enzymes and denature them. So heavy metals can do that. Extreme temperatures, especially high temperatures, and then extreme pH levels can denature enzymes or proteins. If the protein can't refold, the denaturation is permanent, and then the protein will be said to be coagulated, kind of like when you fry an egg. You coagulate the proteins within the egg, and you can't uncook the egg. So we'll talk about factors that affect the rate of reaction. So concentration of either the enzyme or the substrate is something that will affect the rate of reaction. So as you increase either the substrate concentration or the enzyme concentration, the rate of enzyme activity will increase at a pretty steady pace until it plateaus. And that's because there's a chance of more effective collisions, but eventually that rate of reaction will plateau because there won't be any more enzyme or substrate to interact. Temperature, on the other hand, does not have a constant rate of increase and then plateau. The optimal temperature for most enzymes is around 40 degrees Celsius. You think about our body temperature, it's around 37 degrees Celsius, so that kind of adds up. At low temperatures, the rate of enzyme will increase with increasing temperature until it reaches the optimum temperature. Then after that point, it will start to denature, and the rate of reaction will diminish quite drastically, as you can see in this graph. pH, as I said, also affects reaction rate. So if you look here, this is actually showing two different enzymes. So this is the optimal pH for pepsin, which is found in our stomach, so pH 2. Outside of that range, it drops drastically. Same thing with lipase or other enzymes that have a higher pH optimal. You can see that it's around 8 that the pH is optimal, and that would be in our intestines. Outside those optimal temperatures, the enzyme function will sharply decrease. So we'll be doing an investigation exploring the factors that affect enzymes, and we will be using catalase as the enzyme and hydrogen peroxide as the substrate. So catalase is also sometimes called peroxidase. It catalyzes the decomposition of hydrogen peroxide and produces water and oxygen as products. So hydrogen peroxide is a byproduct of some metabolic reactions, and it's toxic to the cell. So you can measure the rate of reaction if you're reacting these enzymes and substrates together by figuring out the volume of oxygen gas that's produced in a given amount of time. So this is one example of a setup that you could do. This is quite complicated. There's much more simple reactions that we could do instead. You can read Cognitive section C1.1.8 to 10 to explore further advice in designing your investigation. So you need to know the difference between intracellular and extracellular enzymes. And again, if you know the root words, that really helps. So intra means within, extra means without or outside of. So intracellular enzymes are built by free ribosomes, and they perform functions within the cell. One example would be the enzymes that are involved in cellular respiration, which we'll be exploring next unit. Extracellular enzymes are built by bound ribosomes on the rough endoplasmic reticulum, and they get shipped out of the cell to perform their functions. So examples of these would be digestive enzymes, so different glands produce the enzymes, and then they are excreted outside of the cell, so into the stomach, into the intestines to do their jobs. So reactions within the body are not just one simple step. They're actually multi-step reactions, so metabolic pathways. So they involve several steps, and they're catalyzed by many enzymes. Some of those pathways will be linear, like glycolysis. That's when we split glucose into 2-pyruvate. We'll explore that next unit. Those paths have a starting substance and an end product. As you start with glucose, you end up with pyruvate. Some pathways, though, are cyclical, like the Krebs cycle, and we'll also explore that next unit. And in these pathways, the final product will be the starting substance. And there'll be a series of reactions that happen, and then at the very end, the last step will be reconverting the final product to be the initial starting substance. So it can go on and on and on. So metabolic pathways are not 100% efficient. Some of the reactions will produce heat, and that will be a lost byproduct. But because we're warm-blooded organisms, that's not such a bad thing because it helps us maintain our body temperature. So an example would be like the aerobic cellular respiration is actually a controlled combustion reaction. Rather than produce an intense amount of heat, we control the steps of the reaction. Some of it is lost as heat still, but the rest of it is converted into ATP, an energy molecule that provides the energy to do the things within the cell. So now we'll talk about inhibition. So competitive inhibition is when the inhibitor is similar in shape to the active site as the substrate. So it will bind instead and block the active site so the substrate can't bind, and then the reaction can't happen. In this type of inhibition, the rate of reaction will eventually reach the maximum rate of the uninhibited enzyme, because when you keep increasing the substrate concentration, eventually there'll be more substrate than inhibitor. So there'll be more effective collisions with the substrate than with the inhibitor. In non-competitive inhibition, the inhibitor binds to the allosteric site. So that's a site that's not the active site. It's away from the active site. And what it does is it causes the enzyme shape to change and changes the shape of the active site, and that prevents the substrate from binding. So in this case, even if you keep increasing the concentration of the substrate, the maximum rate of the reaction will never reach the same as the uninhibited enzyme, because the enzyme shape is altered, and adding more substrate won't fix that. So this is a very common graph that's shown on IB assessments. So it shows the uninhibited, so that's the blue one here. That would be when there's no inhibitor involved. The competitive inhibitor, so as you increase the substrate, the rate of reaction increases, and eventually it will actually match up with the uninhibited. And then the non-competitive inhibitor never can reach, because wherever the inhibitor bonded to, it changed the shape of the enzyme, so it can never bind. So then you end up with only some of the enzymes able to bind, so it will never reach that maximum rate of reaction. So again, we're supposed to look at practical examples of inhibition. So one example would be statins. Statins are drugs that are used to lower cholesterol levels. And what they do is they bind competitively to an enzyme early in the pathway of cholesterol production. So it stops this enzyme right here, right at the beginning. So acetyl-CoA is converted to HMG-CoA. And then all these other reactions happen, but now can't, because the statin inhibits here, which means that some of these other materials aren't produced either. So coenzyme Q10 wouldn't be produced, squalene, glutathione, peroxidase, and so on. But it will prevent the production of cholesterol, which is the end goal. But cholesterol is also used for building hormones and antioxidants. So statins aren't perfect because they actually will prevent some of these other necessary molecules or reduce the production of them anyhow. And product inhibition is when the final product inhibits the initial enzyme, and it's often in a form of non-competitive inhibition. So an example would be isoleucine, which is an amino acid, inhibiting the pathway that converts threonine, another amino acid, into isoleucine. So once you have enough of this amino acid, it will go and bind to the allosteric site and prevent the conversion of more of the threonine to isoleucine. You can see that there's many steps in the process, but it's always inhibiting the first step so that the pathway doesn't begin in the first place. Mechanism-based inhibition is when an inhibitor irreversibly binds to the active site by covalently bonding to it, and that will render the enzyme permanently damaged. And we'll look at two examples. AZT, or AZT, is an antiretroviral drug that's used to treat HIV, and then penicillin is an antibiotic. If we look in depth at the penicillin, it inhibits an enzyme called transpeptidase, which is an enzyme that maintains the cell wall. By inhibiting the enzyme, the cell wall will burst open and the bacterial cell dies because it can't keep its contents within. And then finally, one job for you to do is to read the enzyme inhibition section in Cognity, so see 1.1.14 to 17, and complete the activity on both antifreeze poisoning and the activity on PETAs. And that will prepare you for our upcoming assessment.